Okay, good morning and welcome to the AI and Digital Health session. Hopefully you've had a good morning so far and are excited to join this session. I would like to introduce our session leader to you, Jeroen Maas. Jeroen Maas has a strategic role in driving innovation and business development in the health sector, with a focus on the use of data technology and technology in supporting health at the Amsterdam Economic Board. Fascinated by the complexities of this challenge, Jeroen works with the Amsterdam Economic Board's leading CEOs, scientists, and policymakers to promote innovation with the ultimate aim to gain 2 healthy years for every inhabitant of the metropolitan area of Amsterdam. Jeroen, I will leave a further introduction to you. Welcome, and the floor is yours.
Many thanks, Elske. Thank you for this introduction. And hello, everyone. I miss you already. It's so strange to start this talking into a camera and not seeing you live. So I'm really looking forward to next year's Innovation for Health and see you all in real life. But welcome to this great session on artificial intelligence and digital health. As Elke said, I work for the Amsterdam Economic Board, but also on our regional program, Smart Health Amsterdam. And that is all about data sciences and artificial intelligence and health. And for those of you who were present this morning and heard Ron Fouché talk about pandemic preparedness for a next pandemic from a vaccine perspective, let me take just a few seconds to give you an example of pandemic preparedness that we do in the region using artificial intelligence. It's, by the way, not just in the region of Amsterdam. It was in collaboration with Leiden and Nijmegen. And what was done was using real-world data, develop an algorithm to identify those people most at risk of severe COVID-19. So those people who you would like to vaccinate first in order to get out of a lockdown sooner. But that same system can also be used as an early warning system for any next pandemic. And I'm excited about what AI can bring to health and how we can use that to further health. And that's why I'm really excited to have 3 great speakers in this talk. Maybe, first of all, rules of engagement. Please do ask questions in the live chat. We'll have 3 talks, 1 or 2 questions after each talk, and then a short but intense 10-minute discussion afterwards based on your questions. You can ask them in the live Q&A. We'll monitor that. And then we start off with sort of setting the scene by Ruben Oosterbrugge from Philips. Ruben is a cardiologist and now a global program lead value-based care at Philips. He'll sketch the scene, the broad landscape, and after that we'll go more into detail into what AI can bring in the immuno-oncology. So this session is a great preparation for the 11:30 session on immuno-oncology. But first, Ruben, I give you the floor to explain to us what you, Philips, are doing with AI in healthcare. Ruben, you're still muted. I'm not sure if it's me, but I can't hear anything, Ruben.
Can you try to mute and unmute again? No. Let's see. How about now?
Now?
Yeah.
Yes.
Yeah. Okay. Okay. Yeah. Okay. So I'll put my screen on. Can you also see my screen now?
Yeah. Yes, you can see it.
You can both hear me and see the slides. Okay. So thanks very much, Jeroen, for the intro, and also to the Innovation for Health organization for the invitation to speak here. And I would really like to, in this session, to set the stage on data and AI. And yeah, I'll just start. So to start off with, I would like to mention a couple of trends in healthcare that are driving the need for innovation. Actually, we know that chronic diseases are on the rise worldwide. The prevalence of those keep on rising. We get older, but we also have more chronic diseases. At the same time, healthcare systems are under huge pressures. There's an increased transparency and increased need to deliver better outcomes, but at the same time, the amount of money that is spent on healthcare is constantly increasing. If you look at those graphs of the percentage of healthcare costs as a percentage of the gross domestic product, that keeps on rising. In the US, up to 18% now, whereas in the Netherlands and other Western countries, it's up to 12%. So that could threaten other parts of the economy, but we should be aware of the costs of the healthcare systems. And another trend is that people actually are increasingly wanting to take control of their own health. So patients are also normal human beings, I would say, and it means that they are also consumers. So in a preventive care space or secondary prevention, patients and consumers want to take more control. And actually, digital transformation is then actually a driving force behind all of that. There's an increased focus on healthcare data and how do we actually use it in the best way. And that's where we at Philips think that AI comes in. So how do we believe that AI can help? Well, we think that artificial intelligence uses this data science by people and inspired by intelligent behavior to create systems and solutions that can sense, reason, act, and adapt to a system repetitive tasks. We will go into that a bit more later, but it will be good to also put in our North Star. It's not necessarily only our North Star, but I think it's reflective of how people in the healthcare space think, especially when you think about value-based care. That's my current role at the company. So this is really the different aspects of the quadruple aim that I speak to a lot. So first of all, better health outcomes. So that means better health outcomes, clinically meaningful outcomes to the patients. And of course, those are different in different disease areas. And in order to make sure that we adopt the right outcome measurements, we have a partnership with ICHOM, the International Consortium of Health Outcome Measurements, that provide us with standardized health outcome measurements that are acceptable and really resonate with clinicians. Then, of course, there's the improved patient experience, and there's two sides of that. There's experience in the hospital, how patients experience their care and how pleasantly they experience that, but also more formal way of measurement. So that's the patient-reported outcome measurements or PROMs. We really make sure that we, where we can for our solutions and products that we offer, that we really have those measured. At the same time, we try to do all that with the spirit of not increasing cost of care. So especially in the secondary prevention space, or if you think about prevention, that there's really an important role to play there. And the fourth one of the quadruple aim, which is of course a triple aim, extended with the fourth one, which is staff experience. We really try to make sure that also the staff has a good experience, and that's important because we will run actually out of staff before we will run out of money. So it's really, when I joined Philips 1.5 years ago, I think it's great that one is also in our North Star, and this is really truly in our entire mission and vision of the company. So what we try to do, if we talk about data, to do it along the entire healthcare continuum. So at Philips, I think we are a unique company because we really do it from healthy living, I would say. We have the strong base traditionally when we were still a company that, well, we still make like grooming products, kitchen appliances. Those are actually some of them are being divested, but we have a strong presence as Philips in people's lives. And we try to leverage it now with our new vision and mission in healthcare. So we really try to do personalized lifestyle support. And then if you go one step further, targeted prevention, and then the diagnosis and treatment part, which is more the traditional part of the hospital, is where actually also data and AI plays an important role. Later on, that would go into proactive patient monitoring, trying to prevent patients coming back to the hospital. And that's both good from an outcomes perspective, so patients are doing better, having better health outcomes, but at the same time, it prevents that a lot of high costs are being incurred. So from a value-based care perspective, also the second part is very important. So we really think that AI should be combined with clinical and domain expertise. So of course, you need data. That's a very important aspect, and we'll get to it a little bit how what could be potential hurdles to really adopt data and AI strategies. But if you combine it with artificial intelligence, but really to combine it with the deep clinical knowledge of the physicians and other healthcare professionals, then you can create properly done AI-enabled solutions. So we really make sure that it empowers the people that use them, that it's properly adapted to the context that they're working in, and then integrated in the daily workflow of the healthcare professionals in a seamless way. And of course, when you talk about data and AI, it's very important to make sure that you do it in a responsible way. So that's why we came up with our data and AI principles. To have a more elaborate look, you can follow the link or Google find it on our website. But the most important principles are that it's secure, that we adhere to all the privacy regulations that exist, and to really make sure that it actually benefits our customers and the customers of our customers, which are the patients. And then, of course, by doing that, also the entire society. Our AI principles are related to that as well. Everything should be focused on improving health and well-being. There should be human oversight. As I tried to explain in the previous slide, AI only becomes valuable if it's like combined with the human oversight and interaction with it. So that's a very important principle. The robustness, so we make sure that it's actually valid and tested. We want to make sure that it's fair and that we do not have biases implemented in the AI algorithms. And we want to make sure that everything is transparent. And again, there it's also important that we really show and validate the AI principles that we have, and we make clear that where we actually apply AI technology in our solutions. So as an example, I'm still an academic doctor, so I went to the literature and I came into these very nice articles, and I can highly recommend to read them, look them up, and really think about how clinicians will or will not adopt AI and what are the problems there. So I summarize that in this overview slide that follows now. So what are the hurdles in the adoption of AI and machine learning? Well, first of all, it is very much a behavioral thing. As you know, people, but also doctors are people. It's always very hard to change the way that you actually behave or the way that you work on a day-to-day basis. That counts for doctors, but also for patients. If you think about medication adherence, for instance, only 70% of patients that have a prescribed drug actually fill their prescription. So go to the pharmacy, get the drug. And then of those patients, 70% adhere to their medication. So take it in a proper way. So that means that only half of the patients that have been prescribed a certain medication actually take the medication that they want to, but that they should get. So that's just an example of how behavior is actually really much a central thing in changing the way you work. But more specifically to AI and machine learning, the quality of the data is very important. Of course, garbage in is garbage out. So we need to make sure that, of course, there's the input of the data is of high quality. So EMRs play an important role, but also sometimes logistical or financial data systems of hospitals. And then we immediately go to the interoperability of data systems. So that's the second hurdle. First of all, if you have good data input, then you should actually make sure that all of the data is in an easy way interoperable and taken out of the systems to actually make use in a meaningful way of AI, but also to adhere to data registry requirements. The third one is unfamiliarity. It has to do a little bit with the behavioral part of it, but doctors really would like to know how the AI algorithm is telling them what to do. What is the methods behind it? Why is decision support that is enabled by AI actually giving that recommendation? And whereas doctors are usually trained in normal statistical models, methods like regression modeling, it can be that they are still a little bit unfamiliar with AI and machine learning. And that also has to do with evidence-based. Of course, the world of medicine that we nowadays have is very much evidence-based. However, that means that you also, for AI algorithms and solutions, you have to show that it actually improves outcomes for patients. So that's really important, and that can, if you do that in a proper way, that can drive adoptions for physicians and other healthcare professionals. An important other aspect is to realize that the insights that AI creates are actually actionable. If there's a recommendation that is just there and doesn't make really sense to change your your treatment regimen, that will actually not be adopted that quickly. So if you do a study showing that your AI algorithm is improving outcomes, you should also make sure that it's done in a way that the algorithm is actionable. And then, of course, another hurdle is the approval processes. The FDA, but also other regulatory bodies in the world, are thinking about how to make proper regulatory processes to approve AI algorithms. So there also needs to be a little bit more improvement to adopt all of this AI and data part. So these are the things that we as a company really take into account when we think about AI improvements. And I hope that sets the stage for the meeting or the session that we have today. To conclude, AI represents a dramatic opportunity to improve healthcare by turning data into actionable insights that enable more precise and personalized care across the healthcare continuum. Many point solutions already exist, but the next step is really to integrate them in a seamless way for a complete disease-focused solution. Again, data and AI principles are essential to acceptance, and successful adoption in clinical practice requires solving several hurdles that I alluded to in the previous slide. Thanks very much, and I hope that we can have a little bit of a discussion.
Many thanks, Ruben. I think that was a very clear oversight, and I heard some magic words. You said human oversight. What comes out needs to be clinically relevant, but also clinically actionable. Otherwise, it will never be adopted. So how do you make sure, as Philips, that whatever you produce or whatever you bring to your customers actually is clinically actionable, and how do you make sure that there is human oversight?
Yeah. So the solutions that we develop very much are created together with customers. Of course, we listen to their input. So that's how we make sure that they will be able to adopt it. And the other real important part is that we do proper studies to show that we actually can improve outcomes. That can be in a smaller study, but sometimes it can be larger studies.
Yeah. Clear. Thank you for your answer. There are more questions, but we do have two more very interesting talks. So first, I'd like to go there, and then we have more of a discussion afterwards. Do put your questions in the Q&A when you have them. While we go to, I'll say thank you once again, Ruben, and we go to Cedric Bogaert. Cedric Bogaert is a bioengineer and co-founder of MyNeo. And MyNeo is a computational neopeptide platform to discover new immunogenic targets. And yeah, I think you, Cedric, are the person to explain to us what that really means and what you are doing. Floor is yours.
Okay, thank you, Jeroen, for that introduction. Thanks as well, Ruben, for sharing your vision on how Philips' perspective is on the AI domain. I'll go into a bit of a more scientific approach when I'm discussing my subject, in which we'll be talking about how to match mutanomes of tumor material to improve the immune responses. I'll first start with a really basic background and then go deeper into different aspects of it. And how that can help to improve clinical benefits and to, in the end, improve clinical responses of patients. I think you're all very familiar with my following slide. It's the famous Hanahan picture that shows that a cancer cell is actually being impaired by mutations in certain key processes that allow a normal cell to be transformed into a tumor cell. We're also, because of the sequencing technologies, we have become greatly aware that these key mutations in a cancer cell are not the only mutations out there. There's a complete set, for some tumor cells, it's up to thousands of mutations that are present in the cell that are very interesting to study because these set of mutations, if they are indeed clinically actionable, and they can guide us towards better treatments of patients, they can guide us to lead to certain inclusion criteria, and other kinds of general responses. But next to that, next to those clinical parameters that we can deduce from the mutational landscape of the tumor, there's also a key biological process that's involved in these mutations and why they're interesting on their own already for us. Because any mutation in a cancer cell will lead to mutant peptides being presented on the surface of cancer cells. And these mutant peptides are unknown to the immune system. And as such, the immune system can be targeted against these mutations, against these mutant peptides, to elicit an immune response and to actually eliminate cancer cells. And that's shown here, the biological principle. A mutation in a cancer cell will lead to mutant peptides, and an immune system is capable of recognizing that in a healthy immune system, in a healthy individual, and thus eliminating the cancer cell before they really turn into a full-grown tumor. Certain times, this process does not take place, which then leads to a full-blown tumor. And immunotherapy then tries to actually reactivate the immune system, against these tumor-specific peptides, the so-called neoantigens indeed, to really elicit an immune response against the tumor and to make the patients healthy again. And it's a very elegant process. Immunotherapy, it uses the patient's own immune system. You don't really inject something that's toxic on its own, like a chemo. So it seems rather good in its process on its own. And indeed, the results in the past decade for certain patient groups have turned out to be very effective. And you have certain, for example, melanoma groups that now have up to 40% increase in survival rates. There's far less relapse than with other kinds of therapeutics. And then there's very low side effects. The main thing is that, of course, immunotherapy, it tries to induce immune cells that are specifically against the neoantigens on the tumor cell. And there's also the key challenge there, because actually, when you're looking at the current form of immunotherapies, only 30% of the patients actually respond to them directly. And there's still a large group of patients with cold tumors, I'll get back to that later, that are not that responding, where the immune system is not directly capable of activating its neoantigen-specific immune cells and thus directing an immune cell response against the tumor cells. Indeed, when we're looking at the different immunotherapy forms, they are all directly dependent on how well they are able to induce neoantigen-specific immune cells. And when we're looking at cancer vaccines and adoptive cell transfers, These are really dependent on which target are you using for these therapies. So which targets do you identify on the tumor cells to be present where you want to lead the immune system against? But also other types of immunotherapy. For example, the most well-known immune checkpoint inhibitor therapy. It's been shown in the past years that the response and the efficacy of these therapies is largely dependent on the neoantigen atmosphere, or the neoantigen repertoire that they are induced against. So neoantigen screening can, in that case, be used to measure the efficacy or to predict the efficacy of an immunotherapy. So they are all largely dependent on how well they're able to induce immune responses against these neoantigens. So that means that, of course, the identification of these neoantigens, that's something crucial. When we're considering different therapies, it's essential that we find the right targets to induce an immune response against. Irrelevant target identification can not only overload and saturate an immune system, which of course decreases the efficacy of the overall therapy. An incorrect identification can even lead to immune-related adverse effects, with all the negative effects that come from that. So we're looking at the precise identification. It's a crucial, but it's also very challenging, because when we're considering the complete landscape of mutations of a tumor, only a small subset of those mutations are actually responsible for delivering clinical benefits. Also, for certain tumor types, when we're considering, for example, the cold tumors, colorectal cancers, for example, they don't have a lot of mutations. So there, the challenge really is, how can you identify these mutations? And how can you, if a healthy cell is that similar to a tumor cell, how can you identify still the small differences that are there? And then from that, select the differences that are actually capable of delivering clinical benefits once they are targeted. The targets themselves, these neoantigens, they're often patient-specific as well, which then leads to the issue, of course, that you need to have a well-integrated clinical flow that for each patient specifically is able to identify these mutations and direct the therapy specifically against these. So these personalized flows are something that are being now developed in the past years, but that still requires some regulatory authorities to change their mindset as well. And in the end, you're already hearing from my speech, of course, and also by the topic of this talk and this webinar, that lab assays are not going to cut it. And they're not going to be able to, for each patient or for each patient group, to massively screen thousands of targets to identify the ones that are actually delivering clinical benefits. What is going to help it? Well, it's going to be data-driven predictions. In the past decades, there's been a rapid uprise of computational pipelines that allow to screen the tumor surface, the neoantigens that are present on these tumor cells, and that are direct immunotherapies against it. There's many computational pipelines out there. On the slide on the right, you can see from a review a nice overview of the different open source tools or wireframes that are out there that can be used to go from sequencing data onto what are the targets on the surface. I have some more detailed overviews of these different pipelines and the differences that are existing within these pipelines in the rest of the deck, but I can always share them afterwards. The main essence is they're all based on the same wireframe. They're all using tumor tissue material and healthy blood material or healthy tissue material, do the comparison between the two to identify the targets that are present on the tumor cells. But as I mentioned before, the list of mutations that is present within a tumor can get very extensive, and there's lots of mutations that are present. So the main issue then becomes, how do you distinguish between the differences that are relevant and the differences that are actually just junk material that are not really actionable and that will not deliver any clinical measurable responses? And there's different tools that are used for that. When you're looking first on the first step, the essence is to select only the sources of variation that are present in each cancer cell, so both all tumor metastases and all clones, and that are unlikely to be reversed via immune escape. Next step is to identify and to select the peptides that are actually presented on all of these cancer cells, and that are only presented on these cancer cells and not on the healthy cells. Even a step beyond that is to not only look at these presentations, but also look at which of these presented epitopes, which of these neoantigens, are capable of inducing an immune response. So each peptide is evaluated based on its presentation capability, based on its immunogenicity and other similar values like that. And then lastly, once we have identified a set of specific peptides that are present on tumor cells within the complete tumor biopsy material, it's of the essence to optimally combine these different epitopes to create a vaccine construct that actually allows to elicit a broad and a long-lasting immune response against the tumor material. And there's many sources, and many in the past 2 or 3 years mainly, there's been novel algorithms that have been popping up here and there, mainly directed at how can we use artificial intelligence to better make these predictions, to better accommodate the biological processes that we're trying to approximate. There's some graphs here. Left on top, you can see a machine learning algorithm that's been developed to better identify sources of variation in tumor sequencing material. These sources of variation can be neo-isoforms, can be certain introns that have been shown to be retented in tumor material and not in healthy material, but also post-translational modifications are all alterations that are known to be occurring in tumor material, but that can only be really approximated via machine learning algorithms. On the top right, you can see the increased efficacy that has been reached via machine learning algorithm that looks at the issue of presentation, which peptides will be presented for which HLA alleles and which will not be. Bottom left, you can see immunogenicity predictions, which peptides or which antigens will be immunogenic and which will not be. And if a complete protein is deemed to be immunogenic, which peptide within that protein is actually responsible for that. And all the set of these algorithms has actually improved the target selection procedures for clinical benefit with a factor of up to 10. That means that actually now we're up to a factor 10 better able to distinguish which targets will deliver clinical benefits and which will not be able to do that. To wrap up, I feel forced to quickly disclose as well myNEO, the company itself. We're a team of academics and entrepreneurs. We aim at really at improving immunotherapies and broadening their application fields. Sadly, we've rapidly grown over the past year, so that's why on the left you don't see a group picture of myNEO, but you still see the individual square pictures. On the right, you can see what's been done with the technology. We've used the technology to enter new collaborations, new clinical development pipelines to really bring these targets to the clinic and aid the patients with better immunotherapies. In these kind of collaborations, we usually out-license our technology or computational technology on the targets, but we're open for all kind of new opportunities as well to really bring these targets to the clinics. Lastly, then, I'm happy to announce that we are true to our promise of continuously investing in innovative ideas, which is obviously necessary for data science and for machine learning. So we are dedicating part of our team to continuously doing new exploratory projects. Therefore, we're willing to use our technology for new project ideas that are coming from the general public as well. So if any of you thinks of a new project that could use the technology to evaluate the immunogenicity or presentation or any kind of data processing on antigens or on epitopes or certain peptide fragments, certain vaccine constructs, we're willing to use our technology and the operational team to fulfill your project once your project gets selected. So just submit your project on the myNEO website, and then we'll be able to help you with your neoantigens, with your epitopes, with your data processing that's required for that. That was it from my end. Feel free to ask any questions on our technology if necessary.
Many thanks, Cedric. Great presentation, and I like the way you finish it with a question to the audience. We will go on to the next presentation now immediately to have time for questions afterwards, but for you, I'd like to give you the time to think on this question that I'd like to pose afterwards. You present a gift to the audience, so I think it would be only fair to think about what would you like to get from the audience. How could the audience help you develop this company? But that'll be for a question for afterwards. First, and thanks again, Cedric. But first we'll go to Evangelos Kanoulas. Hi, Evangelos.
Hi.
Evangelos, you're a professor in data sciences at the University of Amsterdam and co-owner of Elegon AI. And that's what you're going to talk about Elegon AI and how you use data sciences, artificial intelligence to match patients with the right treatment. And also, again, in immunology. So please, the floor is yours.
Thank you very much. Thank you very much, Jeroen, for the introduction, and thank you very much for the invitation and the chance to be here and talk to all of you. I think it was very, very interesting to listen to the previous speakers, Ruben's talk and the discussion about clinical validity of the AI solutions, and a good fit to the current flow for uptake of these solutions, but also centric with this work on genome sequencing. And what I would like to talk to you about in the next 10 minutes is Elegon AI and our dedication to also fight cancer by personalizing in immunotherapy, but from a complementary perspective to what Cendric presented, from the perspective of examining the pathology of tumor. So, as you are all, of course, aware of, immunotherapy has brought a revolution to cancer treatment. And this revolution, of course, is due to the fact that it can produce durable responses with minimal toxicities. Nevertheless, what it is not great is the fact that not all patients can benefit from immunotherapy. So right now, what we have is about 15% of responsiveness. And this means, of course, that 85% of patients that are given immunotherapy treatment do not respond to that, which has two big effects. One, it has a cost effect, which in Europe it's about 8 billion euros of unnecessary cost. But also it has humanitarian effects or ethical effects, so to speak, because valuable time is lost. Side effects like the ones that Cendric also mentioned still appear. And this is all about the false positives. So people that take the immunotherapy, but it doesn't work for them. There is also the false negatives and the missed opportunity for those that are not selected for the treatment. So what we do at Elegon AI is what is our goal, our key goal, is to select the right patients for immunotherapy as soon as possible. Now, research tells us that there is a recent proposal of quantifying the potential of response to immunotherapy by what is called an immunogram, a cancer immunogram. And this cancer immunogram comprises of certain parameters that are different to each patient, parameters that we at Elegon, so some of these parameters, if you see, are parameters of the gene, of the genomics, and you can capture through sequencing. But many of these parameters actually depend and can be quantified by the tumor microenvironment. So you're, of course, very well aware that the journey of a patient starts at the moment of the diagnosis. And at that moment, a biopsy is taken from the patient, which is stained and analyzed through microscopy by pathologists. And pathologists are the natural leaders in this sense in precision medicine, because they can look into these biopsies, and they can specify certain phenotypes, certain geometrical structures of cells that they have been connected to treatment response, to immunotherapy treatment response. And now here starts the problem, and the problem is that these biopsies that can be digitized, and you can look at them on your screen, they look like satellite images. Okay, so they look like something like the background picture, okay, for a particular staining. And I'm pretty sure you have all seen this kind of picture. So you can think of this as Europe, right, in a satellite image. And what a pathologist has to do is to count, for example, 2-story floors in the entire Europe. And that 2-story floors can tell something about treatment, right? So basically, the pathology has to zoom in, right? And in this humongous picture. And this in the middle is what the pathologist can see. So it also requires a lot of training from a scarce resource, which is the pathologist, to identify the different type of cells that in there, figure out the geometry, figure out their distances in order to make a call about the possibility that immunotherapy will work, right? What is happening at the very, very deep level is this picture at the left-hand side, at the bottom, where, all these individual cells, are there and can be, in principle, should be recognizable, right? And again, don't forget that the pathologist has to do this in principle if we want to be very accurate for the entire image. Now, this is impossible. This is human impossible. This is where Elogon comes in. So Elogon, essentially, what we do is we develop deep learning technology, deep neural networks, which, if many of you are familiar with, has revolutionized as well the field of artificial intelligence the last 10 years with amazing achievements. So with our deep neural network technology and something that we call sparse learning, which we have invented, we are able, with limited training data from the pathologist. So what is training data in artificial intelligence is a human showing to the machine how to do the job. Okay, now with limited and even noisy such training data, our algorithms can learn with an accuracy that is as good as a pathologist's accuracy. And if we also consider the interpathology variability, it's even more robust. To recognize these different cells, but also to recognize interesting parameters in there. And this for us, for example, tumor-infiltrated lymphocytes, which, for instance, has been proven to correlate well with treatment outcome. Now, this is only the beginning. What we want to do eventually is to have an integrated solution where pathology plays a role, genomics plays a role, the patient record plays a role. So we're at the phase 1 right now. This is our long-term goal. And where we are in this mid-term goal, so in the pathology side, right now we're developing our AI-driven software to identify 3 of the most important biomarkers. This is TILS, PD-L1, and tumor burden, mutation burden. We already have achieved high accuracy for one of them, and we're working on the other 2. Of course, as Ruben said, we're looking at the MDR roadmap, right? So we can get the CE mark, so we can commercially sell our solution. And clearly, we want to also do. We are working already on the clinical validation of our solution. For now, we're looking to participate in clinical trials, so to bring our algorithm, the part of it that is developed, but soon the other 2 biomarkers, to bring them in clinical trials. We're looking at hospitals, the pathology oncology departments of hospitals, but also pharmaceuticals, because you can imagine that patient selection can very, very well benefit from our solution. Now, are we alone in this? We're not. That's great. Actually, there is, companies start picking up on digital pathology and computational pathology. There isn't many, but there are some very interesting companies that are coming up. And but what we think is that we have we have some aspects that we really think are unique. First of all, our collaboration with the Netherlands Cancer Institute, which is very strong, and we got clinically relevant results already. We have our first proof of concept ready. Second of all, this integrated notion that we have, and this unique AI technology that we have developed. What I think also is important is our team, which is it's quite strong, both from the aspect of AI technology, with 3 AI professors, 2 at the University of Amsterdam, 1 at the Netherlands Cancer Institute. Very, very good software engineers, which we trained as well, both within the university and the company. But also the business aspect of it, which we have started developing quite strongly. Now we have done, of course, our analysis of the benefits. What we need, we obviously need to develop these software solutions even further. We want to develop the business aspect of the company, so we're hiring at the level of the business side, but we also need to further develop data. We need to further develop our computational capabilities, and we're looking for a 3 million investment in the next 2 years to be able to do that, to be able to launch our solutions, get the approvals from regulatory bodies, and go to the market stronger and faster. This is all from me, and I'd like to thank you, and happy to take any questions now or in the common question answering slot.
Many thanks, Evangelos, for this presentation and a very clear call to action and questions to the audience. You asked for partners with clinical trials, right?
Correct.
Yeah. So are you already working with the Dutch Cancer Institute within clinical trials?
Yes.
Do you want
not yet for a clinical trial, no, not yet. So at this point, we're validating with cross-sectorial analysis. So we validate our algorithms not in situ, but retrospectively with data that has been collected. But we're in a very good collaboration. We have very strong collaboration with NKI, and we hope eventually to be able to do that. But we're also looking for other hospitals, because obviously these solutions have to generalize to multiple populations, right?
Yeah, that's clear.
Yeah.
Very clear. Yeah, and I think a very clear message. And there is another question from the audience, from Marco. He asked, do you mainly work with images? And if yes, are you considering using different kinds of data like clinical records, biological samples?
Yes, so currently we're working with images, but we are looking into integrated solutions. So eventually we want to take evidence from electronic health records, other kind of either structured or unstructured, so doctor's notes, but also values from tests or all kinds of data that can be in there. For the time being, we focus on images, and that's the key strong point that we have.
Yeah, I think it's very clear. There's a follow-up question. Someone asks, Marco says, thank you very much for the answer. There's a follow-up question about your slide where you put the different companies and said, what's the difference between what you do and companies such as Radiomics or Thirona?
Yes. So right. So there is okay. There are several differences in general, but in the slide I focus on two things. The first thing that I focus is the oncology focus. So there are companies that they are the Google of oncology or the Google of pathology, and they try to solve all kind of problems, segmenting images, picking up different cells. What we do in that sense is very focused. So we not gonna solve the entire pathology thing. We focus on certain biomarkers that we know from the literature that they are correlated with treatment outcome, and we focus only on those. And what we also want to do is we want to really prove that they are correlated well with this treatment outcome, right? So the data we have is these biopsies and the treatment outcome. So we have this whole pipeline of data, and that's the second strong point, because I think many of these companies do have images, but they don't know what happened to the patient. And our collaboration with NKI helps us ensure that we know what happened to the patient even 5 years after they leave NKI. And the third one is this unique technology that we have compared to existing technology that we believe is better.
Thank you. Many thanks to the one who posed the question. It gave you the opportunity to put his 3 unique selling points here on stage. Can we, Elske, can we have the other speakers back on stage? And for the audience, please do ask your questions in the chat. And Evangelos, you made a bridge back to the first presentation we had from Ruben. You said that we know as a company what happens with the patient. And that's one of our unique selling points. That's what you need when you apply AI to whatever clinical process you're looking at, whether it's within the laboratory or like the space that Ruben is working in, more in the hospitals and patient treatment. So I think that makes it nice and round again. But for Cedric, before I come back to the question I asked you before, and just wondering, the theme of Innovation for Health this year is immunology. Everybody is talking about vaccines. I think there hasn't been a time in history that the general public knew so much about vaccines as they do now. So the generic question that would pop up is, okay, so if you can identify those cell surface proteins, does that pave the way towards a very personalized vaccine to treat cancer?
Definitely, yeah. No, and for the really general public, there's indeed a difference between a therapeutic vaccine and preventive vaccine, and the most known aspect of vaccines is the preventive ones. For therapeutic vaccines, it means you're treating the patient with a vaccine while the tumor is already within the body. And there actually, oftentimes, you need to take an individualized approach. You cannot predict upfront what's going to be in the tumor because it's different for each patient. Even 2 breast cancers are completely different from each other. So oftentimes, if you're very lucky, sometimes you can find targets that are present in around 40% of the patients. Then you can do the off-the-shelf therapeutic approach. You have certain shelves ready with treatments, and then you can just apply based on what mutation is present. But oftentimes you don't have any of these shared neoantigens present in a person, and then you need to go for a personalized approach. And I think when you remember my pipeline slides, certain clinical collaborations that we have really focus on using patients' own material to use vaccination specific for each patient. So around half of these days, I would say around 30% of the companies working within immunotherapy development are also focusing on personalized immunotherapy, because although economically a bit more difficult, regulatory also a bit more difficult, clinically it holds a lot of promise.
Is that the same view you have, Evangelos?
Yeah. So sorry, I missed that part, the last part. Can you repeat that? Yeah.
Sorry. Yeah. Cedric said most of the companies working on immunotherapy and oncology despite the hurdles and regulations that you will have if you really make it personal, are working towards that target because it's so much more clinically relevant. So would that be your approach as well, or are you having a different approach to market development?
No, we are working towards. So if I understand, the point here is that obviously, so it's about regulations and privacy. If I understand it, well, we do work towards the personalized solutions. I think this is the answer for us, at least. At the same time, we look at the ongoing discussion both at the European level with the new MDR, but also at the Dutch level. So there is a bunch of committees at the Ministry of Health that are discussing regulations and requirements for a minimum viable product in this kind of AI-powered solutions. I personally participate in one of these committees, so we're having our eyes there. The use of data is critical for what we develop, and this is where some of the difficulty comes from. MDR, so GDPR is there to protect, obviously, citizens. We need to find some kind of solution for, I think, for companies to keep developing. In the US, companies buy data. I think this is maybe not the right approach. So we're also looking at solutions where data never leave hospitals. We train our algorithms in-house. We never get the data. We never see the data, and so on and so forth.
I'll be happy to take that discussion offline with you, because that's one of the things we're working on. But that's not the topic of this session, so I won't burden you with it now. There's a question from Laura to Ruben. Ruben, in your experience, what is the best way to engage and involve patients and healthcare professionals in the design of digital health solutions? You're still muted.
Again, so now you can hear me, right?
That's a euro, right?
Yeah, now you can hear me now. So I think that's a great question, because it's very important, of course, with the end user in mind to develop something. And the answer is basically, if you can think about when to do that and how to do that. So when to do it is actually very early on, even from the strategy to go into a certain space and then interview healthcare professionals, sometimes also patients, and also in the R&D phases, and check iterations of developments. And how to do that is more both in a more semi-quantitative way, right? With surveys, we use those. Can be anonymized, can be open, but then also following through in qualitative discussions. Ask what the real clinical need is, how they would interact with the solution, how they would like it, so that combination of qualitative in depth with sometimes KOLs in combination with survey of, let's say, a general healthcare professional who works in a maybe not in let's say the top academic centers of the world, but in general practice, clinical practice, that really makes a good combination and gives you input on how it will be adopted or won't be adopted.
Yeah, and that sounds like a very robust approach to really incorporate the end user's view and needs in what you're developing.
Yeah.
We're getting towards the end of this session. Cedric, can I just give you 20 seconds to ask for those things, or to name those things to the audience that you would be helped with to strengthen your company?
I think you're going to get the same answer from any AI-driven technology company. If you're asking what can we help you with, it's data. The answer is always going to be more data. We're looking for data sets where we can find new targets in. So these can be large data sets on tumor sequencing data, But on the other hand, we're also looking for partners that have the technology to bring our targets into the clinical development. So we have some targets, some partners there already, but we're always looking for new partners there as well.
Thank you. I think that's indeed the answer that I hear very often. We need more data, but also we need more customers, because it's very, it's easy almost to show what value you add, but it's hard to get the money out of it. We need to end it here. We're 5 seconds over time. I thank you all for your presentations. I thank the audience for your interest here. And I hope to see you during one of the next sessions at Innovation for Health, and definitely next year live. Thank you very much.
Thank you.
Thank you.
Thank you, Jeroen, for hosting. Thank you.